Hello and welcome to the session. Here we are going to talk about the technique flow cytometry. Flow cytometry is a technique which is used to study cells based upon the interaction of light with cells. When light is incident onto the cells are going to deflect that light. Based upon the pattern of scattering of that light, we can get useful information about the cells. We can have information about the nature and the type of cells, we can count the number of cells, and can have other useful information about cells as well. In this process, we monitor cells while the cells they flow through a liquid. So the term cyto means cells, metry refers to measurement. So in this technique, we are going to study properties of cells. We are going to measure different parameters regarding the cells while the cells are flowing through a liquid. So as you can see here that the term cyto refers to cells, metry refers to measuring. So therefore flow cytometry refers to measurement of cells while the cells are flowing through a liquid. We can get various useful parameters through this technique about the cells. This could be useful in providing information about the number of cells of a particular type. Also, it provides information about the size and shape of the cells. Many a times, we can label the cells by using different types of fluorescent substances. And then by measuring the different types of fluorescence which are produced by these cells, we can get useful information about the nature and the type of those cells. So this is based upon how the cells of a particular type they scatter light. Based upon the type of the cells and the nature of the cells, there will be a unique pattern of scattering of light by each cell type. So in this manner, we can study about the nature and the properties of cells. This flow sheet demonstrates the construction of a flow cytometer. The flow cytometer has a light source which produces a light of a particular wavelength. The wavelength of this light could be selected by using the instrument. The cells are present in a liquid, and through the fluidic system the cells are going to flow. So there is a flow chamber through which the cells are going to move by the flow of the liquid. And then when the cells are going to be in the path of light, they deflect that light, they scatter light. So after the pattern of scattering is monitored by the light detectors, the information is then transduced to a computer. And then by using computer softwares, we can deduce the nature of scattering and the pattern of scattering by a particular type of cells. Different cells belonging to various groups, they can move through the flow chamber and the optical system, and they produce a unique pattern of scattering of light, which is then recorded by the computer software. In this manner, by monitoring the pattern of scattering, we can get useful information. This is a diagrammatic representation showing the construction of a flow cytometer. This is a reservoir which contains a suspension of the cells. The cells are brought through this reservoir by using the fluidic system. The cells are then mixed with another fluid which is called as the sheath fluid. Now the cells are going to be introduced here, and the sheath fluid is going to flow around the cell suspension. When the sheath fluid flows and it moves, it is going to pull along the cells with it. And eventually the cells are going to reach this point, which is called as the point of interrogation. This is the point where the light is going to be incident onto the cells. So the cells are going to pass through this portion, this point of interrogation, and light is going to be incident on those cells. The cells, based upon their properties, based upon their size, density, and other properties, they are going to scatter this light. Scattering of light can take place at a low angle. If the light is scattered at a lower angle, then that light, most of that light is going to move in the forward direction. This is called as forward scatter. If the cells scatter light at greater angles, then that light is going to be scattered onto the sides, and that is called as the side scatter. So each cell type will have a unique pattern of forward scatter and side scatter. So there are these detectors which record forward scatter and side scatter. In this manner, these pattern of scattering are recorded for every cell which is moving through the point of interrogation. So this information is recorded by the instrument and then is transferred to a software on the computer. Here in this animation you can see that the cells are present in this reservoir where the cells are stored, and then the cells are going to be transferred through a liquid. So through this capillary, the cells are going to be transferred here, and then here you can see on the outside of this capillary is the sheath fluid. When the sheath fluid is moving, it is going to pull the cells along with it. As a result of which, the cells which are clustered in this reservoir are going to be separated from one another, and then they will pass through the point of interrogation one by one. So every different type of cell will produce a different pattern of scattering when it moves through the point of interrogation. As a result of this unique pattern of scattering, the type of the cell could be identified. So this is representing what is taking place inside a flow cytometer. In a flow cytometer, there is a stream of cells which are flowing in a liquid, and it is surrounded by the sheath fluid. In this illustration, you can see there are two streams of water. There is this blue colored stream of water which is surrounded by transparent water flowing through the pipe. So this blue colored water which is flowing through, when the rate of flow are adjusted, initially it is showing turbulent flow. And when it is undergoing turbulent flow, shown here, so this stream of blue colored water is not well aligned. But later, after adjusting the rate of flow of both of these streams of water, now this blue colored stream, it focuses in form of a narrow stream. So in this manner, the cells are going to flow through the sheath fluid, and when this sheath fluid is going to move around the cells, it is going to pull the cells along it. In this manner, the cells are going to be separated from one another, and they are going to be brought to the point of interrogation one by one. This is also illustrated here. So here you can see that on the outside is the sheath fluid, and the cells are going to move through here. So when these cells are going to come in contact with the sheath fluid is flowing at a relatively higher rate. So it is going to push the cells in form of a narrow beam, and eventually when the cells are going to reach the point of interrogation, so they form such a narrow stream that one cell at a time is going to come in the path of light. This is also diagrammatically represented here, that the sample is going to flow. The sample contains the cells in it, and then the sheath fluid is going to pull along these cells and is going to pull them in form of a narrow beam. So now the cells are going to be separated from one another, and the flow rates are adjusted in such a manner that one cell at a time is going to pass through the point of interrogation. Now here we have some important terminology regarding flow cytometry. The sheath fluid is the fluid which is going to pull the cells along with it, and it is going to partition the cells in such a manner that one cell at a time is going to move through the point of interrogation. The rate of flow for the sample and the sheath fluid could be adjusted by using the settings on the instrument. The injector is going to transfer the cells. The process of focusing the cells in form of a narrow stream is called as hydrodynamic focusing. When the sheath fluid is flowing around the around the sample which contains the cells are going to be focused in form of a narrow stream in such a manner that one cell at a time is going to move through the point of interrogation. So this phenomenon is called as hydrodynamic focusing. This is done by the fluidics component of the instrument. The fluidics component, it includes the liquids and the associated machinery to pump those liquids through the system. Then the cells reach the point of interrogation. The point of interrogation is the point where the beam of light is incident onto the cells. So the cells, they scatter light. Low angle scatter is called as forward scatter, and a scattering of light at greater angles is called as side scatter. So these scattered lights are collected by detectors. This collectively make the optical system of the instrument. And then the detectors, they detect light, and this information is recorded in a computer software. The software plots the information in form of histograms. So this is the electronics part of the system. This picture again shows the process of hydrodynamic focusing by adjusting the flow of the sheath fluid and the sample fluid. So here in this panel you can see that the pressure and the rate of flow are adjusted in such a manner that the cells are going to be focused in form of a narrow stream, and the cells are going to reach the point of interrogation one after the other. Here, if the pressure and the rate of flow are changed, so that would result in a different pattern, and therefore now many cells can reach the point of interrogation at the same time. So by adjusting the rate of flow and the pressure, the cells, they could be focused at the point of interrogation in such a manner that one cell reaches the point of interrogation at one given instance of time. This is a diagrammatic representation showing the pattern of scattering. So this is a cell, and the light is going to travel in this given direction marked by the arrows. So the light interacts with the cells present in the specimen, and some of the light is going to be scattered at low angles. So this is going to be the forward scatter. And then other beams of light are going to be scattered at greater angles, and this is called as the side scatter. There is a detector in front of the light source which collects the forward scatter lights, and then there are detectors on the side which collect the side scattered light. So here in this illustration, you can see that when the cell is going to move through the point of interrogation. So this is the source of light. So when light is incident onto the cell which is passing through the point of interrogation, the cell scatters that light. Some of that light is scattered at low angle. It travels in the forward direction, but it is scattered at a low angle. This light is collected by this detector here, and this is called as the forward scatter. Some of the light is scattered at a greater angle and it is then collected by another detector. So this light is the side scattered light. This illustration represents the correlation between the size of the cell and the angle of scatter it produces. So here you can see that if a cell of a small size is passing through the point of interrogation, then the angle of scattering is low, as opposed to a cell which has a larger size. So the cells with larger size is going to produce greater angle of forward scatter. In the instrument, corresponding to each typical angle of scattering, it produces a particular voltage. So here in this illustration, on the x-axis is the time and on the y-axis is the voltage. So corresponding to every particular angle of scattering, the instrument generates a particular voltage and in turn is recorded as a particular amount of electrical current. So the recording being done by the instrument is actually in form of a voltage which the instrument produces for every unique pattern of scattering. Here it shows the pattern of scattering and the voltage generated for the forward scatter. The same phenomenon happens for the side scatter as well. And then this information is plotted by using the computer software. So here in this graph, you can see on the x-axis is the intensity of the parameter like fluorescence, angle of scattering, voltage, and on the y-axis is the number of events, that how many times a particular pattern is obtained. Then this information is used to produce a histogram. This is another illustration representing a histogram. So in this manner, we get the histogram for the forward scatter. So you can see that this is the distribution of various different types of cells regarding their forward scatter. In a similar manner, a side scatter histogram is also produced. These two types of histograms could be collectively represented here. In this collective graph, on the x-axis is the forward scatter and on the y-axis is the side scatter. So we put this information, that is this particular type of cells, they produce low forward and side scatter. So these are grouped here. These cells, they produce greater amount of forward as well as side scatter. And this particular cluster of cells produce intermediate forward and side scatter. So by putting these two pieces of information together, the forward and the side scatter, so this produces cluster of cells, and each particular cluster of cell will have a unique pattern of forward and side scatter. So these cells are different from one another in their properties, in their size, in their density, in their morphology, and other parameters. So cells of a similar type are going to be clustered together and they will have a unique pattern of forward and side scatter. This is another illustration representing the histogram for the blood cells. So on the x-axis, the forward scatter is represented and the y-axis shows the side scatter. So here you can see that this particular cluster of cells, they are lymphocytes, monocytes, and then the neutrophils. So each particular type of cell produces a unique pattern of forward and side scatter. Once we have constructed the standards and references, then an unknown specimen can be subjected to flow cytometry, and the pattern of forward and side scatter is recorded for every different cell type. Then, by comparing that experimental with a known standard, by looking at their forward and side scatter angles, so we can describe what type of cells are those. If in the unknown specimen, a particular type of cells, they produce the angle of scattering which are typical for the monocytes, then by comparing it with a reference, we can we can identify those cells as monocytes. So if we have a reference, we have a standard, so we can identify the nature and the type of cells based upon their angle of forward and side scatter. So in order to produce good and reliable cytometry data, sample preparation, instrument settings and preparation, and analysis of data, all of these steps are important. So this is again a quick review of the different parts of the instrument, which includes fluidics, which is which includes the fluids and the associated machinery like pumps and pipes, etc., to transport the fluids. Also, the fluidics is important for carrying out hydrodynamic focusing in such a manner that each cell should pass through the point of interrogation one after the other. And then the optics system, which consists of a light source, lenses, mirrors, and other associated parts to collect and detect that light. And then there is this electronics part, which converts the detected light in form of a potential difference, a particular voltage, and then by using by assigning a particular current for every voltage, so the instrument can transfer this information to the software through the electronics system. The forward scatter provides information about the size of the cell, and the side scatter provides information about the complexity and the density of the cell. In this illustration, you can see that there is a correlation between the size of the cell and the angle of scattering. Larger cells produces greater amount of scattering as compared to smaller cells. So this is also represented here. Based upon the angle of scattering, the instrument generates a particular amount of voltage. So if the size of the cell is small, the instrument produces a less amount of voltage. If the size of the cell is greater, then the voltage produced is higher. So for every particular type of the cell, there is a unique pattern of scattering, and it corresponds to a unique voltage. So based upon the voltage which is generated by the electronics of the system, the nature of the cell is recorded and could be identified. Then this information for both side and forward scatter is collected and then is used to produce a two-dimensional plot. If there are more than two parameters which are going to be studied about the cells, then this could be represented in form of 3D plots as well. There is another very important technique which is related to flow cytometry, that is called as fluorescence activated cell sorting. This is a mechanism which is based upon the principle of flow cytometry, but in addition, it is important for cell sorting, for separating the cells from one another. What happens here is that the cells, they flow through the system, and by using the sheath fluid, the cells are going to be aligned in form of a narrow stream. By using a vibrating mechanism, the stream of cells is split into small droplets in such a manner that each droplet contains one particular cell in it. Now this droplet of cell, it is allowed to move through the point of interrogation where the cell, it produces a particular pattern of forward and side scatter. This is recorded by the instrument, and then the instrument monitors this particular pattern of fluorescence which is produced by the cells, and based upon that there is a system which adds charges to that droplet containing the cell. Based upon the type of the fluorescence or scatter which a particular droplet of cell is producing, the system assigns a charge to that droplet. So the droplets, they may become positive or negative charges as represented in this case. And some cells, those cells which are present in a droplet which is positively charged, when they move through the system, so later they are influenced by these deflection plates. The deflection plates are charged, so the negatively charged deflection plate is going to pull the positively charged droplets towards itself, and the positively charged plate is going to pull the negatively charged droplets of cells towards itself. And in this manner, these droplets containing the cells, they are going to be separated from one another based upon the type of the fluorescence or the nature of the scatter they are going to produce. In this mechanism, the cells are identified from one another. They are labeled by using fluorescent dyes, and the cells are detected by using the pattern of fluorescence which they are producing. And then, based upon that pattern of fluorescence, the cells could be separated from one another. So this mechanism, this process is called as fluorescence activated cell sorting. So in this process, the cells of different types, they are identified. Not only they are identified, they can also be separated from one another by splitting them into small droplets and then assigning a particular charge to each of the droplet containing a unique type of the cells. So this mechanism is called as fluorescence